All right. Welcome everyone to Bio 153, Human Anatomy and Physiology 2. The first chapter we're going to be covering this semester is Chapter 19, Blood. And this is a nice chapter of review from Bio 152 and a nice connection point to continuing on in the sequence to Bio 153. And so how appropriate we have all these red blood cells on the screen here. So, alrighty. So here we go. So taking a look, we're first going to review the physical characteristics of blood. And so why not start off with a nice little picture here of blood samples. So you probably have your yearly physical checkup with your family doctor, and he or she may say, it's time to go get your blood samples done. And I know that's the case for me. My doctor's like, all right, Chris, it's time to go get your blood samples done. And so, you go into the Med Express, maybe you go to Covenant Healthcare, and eventually your name is called and you go to the back room and the phlebotomist comes in and they go ahead and extract your blood. And maybe there's a couple vials they need in order to maybe take a look at your cholesterol or triglycerides or hemoglobin or calcium, or maybe just do a complete CDC. Take a look at your white blood cells, platelets, etc. And so when the blood is taken from you, the color typically will be the scarlet type of red color, and it can be sticky and oblique. The pH of your blood is within a very narrow range of 7.35 to 7.45 with a temperature of 38 Celsius. Taking a look now at the functions of blood, kind of just a broad overview, really. The first critical function is transportation. So your blood is going to be able to transport a variety of items throughout the body, one being nutrients, which is referring to really your diet. What are you putting into your body? So this is really referring to fats, carbohydrates, proteins. And then gases are also transported through the blood too. The case with oxygen, O2, and CO2, carbon dioxide gases. Even metabolic waste products can be transferred through the blood. Maybe you have excess amount of hydrogen ions that have been built up. Or maybe it's a huge increase in urea, which is produced from protein metabolism. Or maybe it's a buildup of creatine or creatinine. A lot of different products that are defined as waste products. And then ultimately, these waste products are going to be eliminated from your body either through urination or defecation. And then you can even have hormones that can be transferred through the blood. Hormones such as insulin or glucagon, which play a very critical role with blood sugar regulation. Or it simply could be movement of hormones through the blood from one organ to another, such as testosterone or estrogen. And the list goes on and on. Another critical function of blood is protection. So the case with blood clotting that platelets will assist with. Or the immune system response. White blood cells play a very critical role with that response. If there's an encounter with bacteria or a virus like COVID-19. And then, of course, overall blood temperature regulation is very critical as well. And this is key, of course, for absorbing and distribution of heat throughout the entire body, and especially to your organs of the body. We're now going to take a look at the major components of whole blood. So blood as a whole is defined as a fluid connective tissue. And when blood is going to go ahead and be withdrawn from you, extracted from you, from the phlebotomist, you can see the image here. The blood is taken and then eventually placed into a vial. And then that vial, that test tube per se, is then transferred into a centrifuge. So this is a big centrifuge. This is a type of apparatus that can be used in the medical field. And of course, something you would see, in chemistry or in biology. And then the vials of blood are put into the centrifuge and spun at a typically high RPM rate. And then the blood is broken down into two major components. Broken into what's known as the plasma, the yellow portion, and then the bottom of the sample of blood, the red portion, is defined as the formed elements. So the plasma, the yellow portion, typically on top in the vial, is made up of 55% of whole blood. The plasma is considered to be a non-living fluid matrix, a good proportion of water, but also a percentage of plasma proteins and other items as well. What divides the plasma from the formed elements is what's known as the buffy coat. So this thin meniscus line right here. The formed elements on the bottom is going to be made up of three important items: leukocytes, and leukocytes are referring to white blood cells, platelets, and erythrocytes, referring to the red blood cells. Here we have an image of blood right here that this sample of blood has been spun down in a centrifuge. And so we're able to see here that light yellow, which is referring to the plasma, and the red here referring to the formed elements. An important term when it comes to terminology, and there'll be lots of terminology in here this semester, is looking at what's known as the hematocrit. Hematocrit. The hematocrit is the overall percentage of total blood volume in a given blood sample. And really targeting in here the red blood cells. So typically, males have a slightly higher percentage of the hematocrit versus females. And you can see the difference is a difference of really 4 percentage points. Ultimately, the difference is because of an overall larger size of body mass with men versus women. So typically men, statistically, if you have 100 men and 100 females, women, there would be a statistical difference there, ultimately, when it comes to overall muscle mass, bones, structurally. So that's how we end up with the difference in the hematocrit. All right, next taking a look at the plasma. What exactly do we have in the plasma? I mentioned that there's water. I mentioned that there's plasma proteins. But there are some other things sometimes that can be found in that plasma as well when we consider blood plasma. And this is typically something that's very important, especially for soldiers that are out on the battlefield. Plasma is critical if they're injured. And especially for patients that are going to be having any type of serious surgery, they're more likely than not going to need plasma. There's even plasma banks that you'll find throughout various communities There's plasma banks in Saginaw and Bay City, Midland, where you can go ahead and donate your plasma, and you will get you'll get money for that if you donate your plasma. And in order to get just simply plasma and not the formed elements, typically there's an electro sieving device that can go ahead and remove the formed elements, and hence you're left with plasma. So here you can see the variety of items that can be found there. There can be some organic waste, granted not exactly beneficial, but that will, that can be removed from the body. But the nutrients definitely come in handy, so that's really important. Especially electrolytes, very important for proper balance of these electrolytes when it comes to normal overall muscular contraction and for your heart to beat and things like that. Respiratory gases, of course, important, especially oxygen here. Hormones, and then plasma proteins are also very critical. So definitely, this is just trying to show you here some of the important items that can be found in the plasma. Okay, so what we're taking a look at here is a microscope slide of a human blood smear. And so this would be a normal slide that you would find. So if you're scanning, your microscope slide, maybe you're in microbiology or wherever it might be, there's a variety of items you'll find. So the platelets, here we have some platelets right here. So here we have kind of this aggregate clump of cells. So this is the platelets that you'll find under the slide. And then we have the erythrocytes, right, our red blood cells. So these are going to be the pink cells here that are biconcave and kind of look like donuts almost. So these are all red blood cells, a lot of red blood cells on the screen. And then you'll also notice a variety of white blood cells. So we're seeing here the monocyte, neutrophils, lymphocytes. So those are 3 white blood cells as an example. The way you'll be able to distinguish a white blood cell from a red blood cell is ultimately the adult white blood cell will have a nucleus, which is the case here, here, and here. In contrast to red blood cells, in their adult form do not have a nucleus. So that definitely you can see that here with all these red blood cells. We're first going to really focus in on understanding red blood cells, erythrocytes. So that's what we're looking at here in this picture. And one of the main important functions of red blood cells is the transportation of respiratory gases. So that's a very important function for RBCs, is the overall respiratory gases. And again, when we're referring to these gases, we are referring to O2, right? Referring to oxygen. So very important gas that we need. And also referring to carbon dioxide. So CO2, right? For carbon dioxide gas. So there's our carbon, and then carbon dioxide. So pretty important. And hence, these red blood cells are also critical because of the fact that they are transporting hemoglobin. And this is a very important protein that we're going to find here on the red blood cell. Very key for transporting these gases, oxygen and carbon dioxide. So now we're taking a very in-depth look at the structure of hemoglobin, really looking at the biochemistry of hemoglobin, this very critical protein. So off the bat, jumping right in, you're going to find that there are two major types of polypeptide chains that make up hemoglobin. On the top, we have our beta chains, so two beta chains, and on the bottom, two alpha chains. So the beta that you see here, these are beta. So this is the Greek notation. So this equals beta. Okay. And down here we have alpha. So that's alpha is Greek. So there we go, alpha. So that's the first letter of the Greek alphabet is alpha. And then you have beta when it comes to Greek. Alpha and then beta. So you have 4 polypeptide chains total, but remember, you're going to have 2 alpha and 2 beta that ultimately equal 4. Now, for each polypeptide chain, in the middle you have a heme group. And then if you take a look at this picture, here we have the heme group, okay? So this is the heme group right here. So this is our heme group. And in the middle of the heme group, you have iron, okay? So this is iron right here, okay? So the Fe refers to iron. Very important mineral that we need. And what's critical about the iron is that this is where the oxygen molecule will bind. So an iron atom in each heme group can bind to 1 oxygen molecule. And when we look at the hemoglobin molecule as a whole right here, each hemoglobin molecule can transport 4 oxygen molecules. And that's because, again, you have 4 polypeptide chains, 2 alpha and 2 beta, and each one has the heme group with the iron in the middle where the overall oxygen will attach. And when you really extrapolate this, one blood cell in your body has roughly 280 million hemoglobin molecules. So that's a lot. And then you think of all the blood cells in your body, of red blood cells, erythrocytes, and wow, that's a large number. So, okay? So definitely really important things to know on this slide. All right, so we're now going to review, and this is an opportunity for you to go back and look at your notes and make sure that you're grasping some of these really important concepts. So it's review time. All right, so our first review question for you to work on as you're working on this at home. 1 oxygen molecule will bind to a blank atom on a heme group. So, which of these would be the correct answer? So we'll give you a couple seconds to look through your choices, and hopefully you select the correct answer. All right, and the correct answer here, hopefully you have selected iron. As iron, letter C, would be the correct answer here. All right, number 2. Each hemoglobin can carry how many molecules of oxygen? So based off of these choices, what would be the correct answer here? All right. And the correct answer would be choice letter B, 4 oxygen molecules. So, very good. Number 3 for review. Which of the following make up the formed elements of blood? So what would be the correct answer here? We think of the formed elements of blood. All right, and the correct answer here, I would probably go with choice letter D. Both A and B would be correct. So the leukocytes and the platelets. Wouldn't be the plasma, right? Because the plasma is referring to the yellow portion here when the blood is spun down the centrifuge. But the formed elements is referring to the platelets and the white blood cells. But also, don't forget, it's also referring to the erythrocytes, right? It's referring to those red blood cells. And the formed elements, of course, is this part right here. So, definitely important concept to know. All right, moving on. Some new terminology here that's really critical for you to know is looking at the term which is known as the hematopoiesis. So a very important process here, the hematopoiesis, referring to blood cell formation. So all blood cells are going to arise, their origin coming from a stem cell. And the name of that stem cell is known as a hemocytoblast. Hemocytoblast. And the location of where we find these stem cells in the human body is the red bone marrow. Okay, the red bone marrow. So the various bones where this would be, this red bone marrow, and when we think of stem cells, would be the overall axial skeleton, the girdles, referring to a lot of the bones in the hip vicinity, and also the ends of the humerus bone, the long bone in the arms, upper arms, and of course the femur. It's the strongest bone in your body. So you've probably seen a lot of terms that are that are indicated as hemo or hemato, and ultimately this is referring to the idea of studying of blood, which is hematology. So hematology is the study of blood. We're now going to take a look at another term, which is known as erythropoiesis. Erythropoiesis. And erythropoiesis, you break down this word, and the first portion, of course, is referring to the idea of red. And poiesis referring to this process. So referring to the genesis of red blood cells, the making of red blood cells. So this starts with, again, your stem cell, which is the hemocytoblast. That's the origin of these red blood cells. And then what will happen is this cell will slightly change and become a committed cell. And this is known as the proerythroblast. So ultimately the cell is committed to becoming a red blood cell, an adult red blood cell. From this process, then we get into the development pathway. So phase 1 here is what's known as the ribosome synthesis. So that's going to be your development of ribosomes. And of course, the ribosome is a very small organelle that you find in the animal cell. Phase 2 of this process is hemoglobin accumulation. So you're going to have an increase in hemoglobin. Hemoglobin is going to be produced. Okay? And ultimately then, phase 3 is going to be the ejection of the nucleus. So literally the nucleus is going to leave the cell. So here we have this nucleus here, and here it's released from the actual cell. And then what you're left with then is a structure known as the reticulocyte. And the reticulocyte ultimately is the precursor to having the mature red blood cell, which of course is the erythrocyte. Okay, so that's what this process is known as, erythropoiesis, ultimately the process of generating adult red blood cells. We're now going to take a look at the regulation process of erythropoiesis and production of red blood cells. So this gets us back to an idea, a term that you would have learned in Bio 152, which is known as homeostasis. So you're probably thinking, oh yeah, I remember that term, homeostasis, right? And homeostasis is ultimately referring to this idea of a stable environment, of equilibrium. And ultimately that there's this idea of this normal range of all your blood chemistry values. And it's important for your body to be in this normal range of red blood cells, and likewise a normal range of triglycerides or cholesterol, whatever it might be. Because if you fall out of the normal range, and maybe there's a deficiency or an excessive amount, both of those are really can be a big, strong negative on the body, and can lead to different diseases and severe effects. So it's very important, again, to have your blood reviewed for, every year, have blood samples taken to make sure, am I in homeostasis for my various blood levels? In this case, we're looking at normal blood oxygen levels. So in this situation, we're looking at where maybe you have an individual that runs into an issue called hypoxia. When you hear the term hypoxia, hypoxia is referring to low oxygen levels. And this can occur for a variety of reasons. Perhaps someone's in hypoxia because they have a decreased RBC count. So maybe their erythrocyte count is very low. Maybe their hemoglobin levels are at a very low amount too, which in fact can lead to the hypoxia. Or maybe there's a decreased amount of oxygen in the body. These are all different scenarios that can lead to hypoxia. Or maybe it's even someone has some form of anemia, some type of red blood cell disease that has led to this. How does the body react to this? We can't stay in the state of hypoxia forever. So what will happen, the kidney, right? This is the kidney right here. The kidney is going to release a very important hormone known as erythropoietin. So erythropoietin. Sometimes you might hear of this hormone abbreviated as EPO. EPO is an abbreviation for erythropoietin. So erythropoietin, again, released by the kidneys in response to hypoxia. And the hormone then will leave the kidney and will go to the bones. And this hormone will actually communicate with the red bone marrow, and it stimulates the red bone marrow. Pretty much tells the red bone marrow, hey, you need to go ahead and produce more red blood cells. So that pretty much gets that process of erythropoiesis going, and ultimately can lead to significant increase in erythropoiesis to increase your RBC count. And then ultimately, you have these red blood cells, RBCs, finally getting back to a normal level. Now, this brings us into the topic of blood doping a little bit here too. So if somebody's doing blood doping, typically it's usually because they're involved in a endurance type of activity. They're running, or they're cross-country skiing or swimming or whatever it might be. And they literally will have blood taken out of them a couple days before the event. And then it's put back into them. And then ultimately that leads to a significant amount of red blood cells. And then they ultimately have the ability to have more oxygen going throughout their body, and this advantage with endurance activities. Now, granted, that's definitely not ethical and banned in many different sporting competitions, of course. All right, we're going to take a look now at the life cycle of red blood cells. So the majority of cells in your body are not the same cells you had at birth. Many of your cells go through this life cycle process where cells are produced they get to the end of their functional lifespan, and then they break down. So that's what we're seeing here with the red blood cells. So ultimately, we can see that red blood cells have a functional time frame, and you can see this down here too, of between 100 and 120 days, ultimately, for adult red blood cells. That's their lifespan, typically, for an average. So when you think about that, for the amount of months per se, we're in the ballpark here of approximately 3 to 4 months. When you think of months 30, 60, 90, 120. So 3 to 4 months is the typical lifespan for red blood cells. Once the red blood cell makes it to the end of the life cycle, it's time for the red blood cell to degrade and break down. Some items will be eliminated from the body. Other items will be recycled and used again in the body. So right up here, we focus in on hemoglobin, which would be an erythrocyte. And this hemoglobin here can be broken down into the globin portion, then to amino acids, and the amino acids can actually be utilized in the body again. So they'll go back into the bloodstream. The heme portion here, however, ultimately can be further broken down into bilirubin, and the heme also ultimately can be iron. So the iron in that can be stored either as the ferritin or hemosiderin. And you can see them over here. These are going to be here the protein iron complexes here. And then with the iron here, these complexes, you have a transport protein known as transferrin, and that can ultimately transport the protein ultimately back to the blood. The bilirubin, however, temporarily then is in your liver. And then this bilirubin is picked up, is transferred through the blood, and eventually then makes its way to the intestine, and eventually leaves through defecation. So you may be asking yourselves, well, where are these red blood cells broken down? Where is that location? So there are 3 major locations where red blood cells are broken down. So 3 locations where red blood cells are broken down. So one being the spleen. That's a major location in your body where red blood cells are broken down. So the spleen, major location. Another major location where red blood cells can be broken down is your liver. So the liver is another major location where red blood cells can be broken down. Another major location, number 3, is going to be the red bone marrow. So the red bone marrow is also a major location where red blood cells are broken down. So I consider those the big 3. Okay? So very important to make sure to know those 3 locations where these red blood cells are being broken down in the body. Moving on, we're going to take a look at some different red blood cell diseases. The first one we're going to take a look at in here today is looking at sickle cell anemia. So that's a very common type of anemia that you maybe have heard about before, perhaps. And when we talk about the term anemia, it's referring to blood that has a very abnormal low oxygen carrying capacity. This is genetically passed down from one generation to the next. And typically, this is usually found in individuals based off their ancestry, individuals typically that are African American or that are of African descent. So, symptoms of someone that may have sickle cell anemia: they're very tired all the time, so very extreme fatigue, shortness of breath, breathing's a problem, maybe even chills or paleness is a possibility. And the red blood cells will be a sickle shaped, indicating low oxygen, because there'll be less binding sites with the cell shaped significantly different, very tough for that oxygen to bind properly to the iron. So this picture kind of shows that here. Now the thing is not all the blood cells are going to be sickle shaped, but you may have a good amount that are. So like this is a sickle shaped red blood cell, here's another one, here's another one here, here's another one here, etc. So we look at this, and here we would have a normal RBC, normal red blood cell, this biconcave circular shape, versus the red blood cell that is more sickle shaped here. Okay, so here we have the sickle RBC. Almost looks like a crescent moon. So there we go. That looks all right. So this gives you an idea here. So definitely be familiar with sickle cell anemia. Another thing to consider, how the cell changes. I mean, ultimately there's, a genetic result, but more on the biochemistry level of sickle cell anemia. When we look at protein structure, if you remember the different protein level structures from Bio 152, right? There was like the primary level, secondary, tertiary, quaternary, etc. If we simply look at the primary level, right? The primary level of protein structure is referring to the sequence of amino acids, typically, for the primary protein structure. And these are all the different amino acids that you'll find here: valine, histidine, leucine. And these are the abbreviation for these amino acids. Ultimately, the change in this primary protein structure level occurs at position 6. So here's position 6 here. So position 6 is glutamic acid. Okay? So this is where this abnormal base substitution takes place, where we go from a normal red blood cell right here, right? This is a normal RBC. So this is normal. So normal RBC, right? Has that circular biconcave shape. The change occurs, genetically speaking, unfortunately, at position 6 in this primary level of protein structure. So glutamic acid, this amino acid then is replaced by a new amino acid known as valine. So right here. This single change alters the shape of the red blood cell from a normal shape to this kind of crescent moon shape. So we go from normal red blood cell to the sickle cell. All because of an abnormal base substitution. So here we have the sickle cell erythrocyte, IBC. Okay? So very important to know. Another type of anemia, definitely a very common one, is the iron deficiency anemia. Iron deficiency anemia. By far the most common type of anemia out there. Ultimately, it's because of very low hemoglobin levels. And there's a variety of causes for this. A major cause is simply the person's diet. So if someone has a very poor diet and they're not eating a well-balanced diet, they're not getting a whole lot of iron in their body, it could lead to them ending up with iron deficiency anemia. And you get a lot of iron from eating a lot of whole grains, from whole grain bread, bagels, cereal. You can get iron in spinach and several other vegetables. And you can get a lot of iron if you eat a lot of meat too. If you like eating liver, there you go, perfect, right? Or you like eating pork or beef or fish, you're getting that iron in you. Another cause, definitely not as common, is the impaired iron absorption. So somebody is struggling to properly absorb iron in their GI tract. And then also if the individual is experiencing blood loss. How can you identify this under a microscope slide? Well, like this arrow is pointing to, typically you'll find red blood cells that are kind of a little bit more pale. And that's because of the lack of hemoglobin iron. So there's going to be a paleness to these cells. The next type of anemia we're going to take a look at here for you today is what's known as pernicious anemia. Pernicious anemia. And this is ultimately caused by a deficiency of vitamin B12. So somebody's not getting enough vitamin B12 in their diet. Another possible cause could be the lack of an intrinsic factor. That would be in the stomach that's needed for the absorption of vitamin B12. Typically for treatment here, some people may just get regular injections of B12. And if you were to identify this under a microscope, typically you'll have red blood cells that are quite large. So that's typically a sign here. So here's some that are a little bigger than the norm, for sure. And sometimes they have a difficulty dividing, mitosis. So we're kind of seeing that a little bit here too. I mentioned blood doping a little bit back when we were talking about the idea of erythropoiesis, tissue hypoxia. And this gentleman's a great example of someone who did a lot of blood doping back in the day, Lance Armstrong. So maybe you remember him. Maybe he was your role model growing up in life. Maybe you're really into biking and stuff. And it was quite amazing. I was in college at the time when he was at his peak, really. And he was doing great, winning the Tour de France 7 times in a row, as you can see here. That's a big biking race over in Western Europe and France and Italy and that vicinity, Spain, etc. But eventually it came out that Lance Armstrong was blood doping, and where that's something that's considered unethical, where he was taking red blood cells. The physician would go ahead and take blood from him and re-inject his blood several days before his big competition, leading to excessive erythropoiesis. And so many red blood cells were in his body. He had huge advantage with more oxygen going to his muscles and advantage in competition. So definitely something that, again, very unethical. He ended up being banned from professional sports. And unfortunately, many athletes have gone the route of blood doping and cheating. And that's been caught in the Olympics too. So very unfortunate scenario here. We're now going to shift gears, and now we're going to talk about white blood cells. We've spent a lot of time looking at red blood cells, but how about we'll spend some time on white blood cells? So leukocytes. Leukocytes, again, the fancy term for white blood cells. And in your body, you're going to have a variety of several different types of white blood cells. And they're broken down into two major categories. So first today we'll take a look at the granulocytes, and then we have the agranulocytes. So the granulocytes are made up of three major types of white blood cells, which are neutrophils, eosinophils, and basophils. And you probably noticed the percentages here too. So like neutrophils, they're the most abundant white blood cell type in your body. You can see the high range here. And then eosinophils and basophils have a significantly smaller percentage. With agranular cytes, there are lymphocytes and monocytes. You can see a pretty high percentage of lymphocytes in contrast to monocytes. And ultimately, to get these percentages, this can be performed by doing what's known as a differential white blood cell count. And this can occur if you were to look at a normal slide of blood. Maybe it's from a patient and you're working with a hematologist in the laboratory. You could look at a sample of blood, and you could go ahead and do just a random count of 100 white blood cells. And, someone's tallying the data, and these are the percentages that you would find. So doesn't that sound like a lot of fun? Working in the microbiology room or the hematology lab, at Covenant or, Ascension St. Mary's or wherever it might be. So imagine all that fun and counting all those cells. What a blast, eh? So what's important to know about these white blood cells is really focusing in on their functions. So that's going to be what's critical as we go through these slides together, is knowing the functions of these white blood cells. So first we're going to focus in on the granular cytes, right? The leukocytes. And the characteristics you'll find with granular cytes is the fact that they have visible cytoplasmic granules. And the shape of the nucleus is multi-lobed. So first we'll take a look at the neutrophil today. Like I mentioned, the neutrophil right here is the most numerous white blood cell in your body. And one of the major functions of neutrophils is the fact that they're phagocytic. So phagocytic meaning that they play a role with the immune system. They can undergo phagocytosis, and they typically go after bacteria, okay? So they'll go after and they'll kill bacteria. But they also can kill viruses if they want, or they could go after deadly fungi as well, or even toxins. All right, next in the middle, we have the eosinophil. So here's our eosinophil right here. So kind of more of red cytoplasmic granules you can see. And one of the major functions of the eosinophil is that they will go after parasites. They can kill parasitic worms if you're exposed to them. And you can definitely get them if you eat raw fish. As much as we love our sushi, well, you're probably okay here in the States. But if you're eating sushi in Vietnam or, China or wherever, I don't know. That's do at your own risk, I guess. Risk assessment, right? So that's a major function of eosinophils. They also can be phagocytic as well. So that's a major function for neutrophils. But eosinophils can be phagocytic too, just so you're aware. And then the rarest of the white blood cells in your blood are the basophil. So here's the basophil here. they have more of a purplish black cytoplasmic granules. And one of their major functions is that they release the overall chemical known as histamine, which can lead to inflammation. So this happens typically when we think of allergens, when someone has an allergy, right? So when somebody has an allergy and they're allergic to something. So an allergy. So someone that might be allergic to poison ivy or stung by multiple bees or hornets or wasps or peanut butter or whatever it might be. So that's a major overall function of the basophil, is releasing the chemical histamine for inflammation. Now, there are other functions as well that your textbook covers too, and so it may not hurt to take a look at that. But these are just some that I'm talking about in here today. We now take a look at the functions of the agranulocytes when we look at white blood cells, leukocytes. Characteristics of agranulocytes: they lack visible cytoplasmic granules, so you may not able to see them under the microscope per se, and the nucleus, typically the shape is different. The shape will be more of a kidney-shaped or even of a sphere, and may even take up most of the interior of the cell. So first we take a look at the lymphocyte. So here is the lymphocyte right here. So you can see here that the overall nucleus takes up most of the interior of the cell, and lymphocytes play a big role with the immune system response. There are two major types of lymphocytes in your body, which are T cells and B cells. T cells have the capability of fighting against virus-infected cells and tumor cells, so cancer cells. So that's a big function, to go after those virus-infected cells. So pretty important, for someone who's battling COVID-19. And then B cells, another type of lymphocyte, ultimately will give rise to plasma cells, and they produce antibodies. So that's pretty important. B cells produce antibodies. And then finally, our last white blood cell type today is the monocyte. This is the largest of the white blood cell types, and a monocyte can undergo phagocytosis. That's the major function of the monocyte, is they're phagocytic, just like a neutrophil. And so that's a major function that they can undergo. They have the capability of developing into macrophages and tissues in the body, and ultimately will go after and kill viruses like COVID-19 and bacterial parasites. But they can go after other forms of bacteria too, and can also go after deadly fungi in the body. So very important to make sure to know the functions of white blood cells. So that's very critical to make sure to know. So definitely make sure to review that. All right, just like we went over previously with the production process of red blood cells, it's also important to know the production process of the white blood cells. And that is known as leukopoiesis. Okay? So leukopoiesis. Very important to know that. Production of white blood cells. So this all starts again with our common stem cell once again, which is known as the hemocytoblast, right? That's our common stem cell. Just like with the red blood cells we mentioned. And from that, the hemocytoblast, the stem cell, will evolve into two major types of stem cells, into the lymphoid stem cell and the myeloid stem cell. The lymphoid stem cell ultimately will become lymphocytes. And the myeloid stem cell will ultimately evolve into eosinophils, basophils, neutrophils, and monocytes. So, those are the important things to really focus in on this slide. You don't necessarily need to memorize all the terminology of the committed cell and developmental pathways unless you're very much into hematology, but these are the things that you need to focus in on to know. It's also important to be aware of various white blood cell disorders and diseases. And so we take a look here today briefly talking about leukemia. Leukemia. So leukemia is a form of cancer that causes the number of white blood cells to increase. So their number increases. But unfortunately, those cells stay in an immature stage and ultimately are non-functional in the body. There are many different forms of leukemia. The two major categories are the acute leukemia and the chronic leukemia. So someone that has acute leukemia, typically this cancer will target children. It's very severe. And anytime you hear the word acute, anytime you hear the word acute, when we talk about diseases, acute refers to a disease that often moves pretty fast. It's pretty fast, short term, okay? So pretty severe, more short term, usually lethal. So acute, short term. In contrast to someone that might have a chronic leukemia, anytime you hear of the term chronic, I mean, it's definitely something you need to pay attention to, but chronic means you can live with it for a very long time, years, decades, but important to have that particular disease or situation under control. So chronic typically is more long-term. Acute, short-term, acting fast, versus the chronic, which is more long-term. And someone can live with this various disease as long as the medication, you're taking that and it's controlled. So chronic leukemia, typically found more in the elderly and older people. And they can live with this for a number of years, possibly, as long as they're taking their proper medication. They're taking their chemotherapy, drugs, maybe they're taking radiation, maybe they're getting steroids, whatever it might be. There are many symptoms of leukemia, as you can see in this picture down here. Someone could show all these symptoms or maybe couple symptoms. So just something to keep in mind. And I go over this because very key. Many of you are going into nursing, and you might be working on a cancer unit someday. Here are a couple types of leukemia here that can be acute or chronic. One type we have here, this is known as the lymphocytic leukemia. So this is referred to an increase in lymphocytes. So here we have a picture of many lymphocytes here. Unfortunately, they never make it to their adult stage. And this type of cancer, again, acute, obviously more severe versus chronic. Ultimately, the person can die from having lots and lots of infections. And the body just can't keep up with controlling them. Another type of leukemia is the myelocytic, granulocytic leukemia type. Again, this could be chronic or chronic, acute or chronic. And again, this picture is showing here Someone that has this, it's usually going to be a significant increase in eosinophils, basophils, or neutrophils. And the picture is pointing to some of these various cells here that, again, are kind of stuck in this intermediate stage of development. So we leave white blood cells, and the last cell type that we need to cover today is looking at our platelets. So platelets. The major function of platelets is for blood clotting. That's their major function, is for blood clotting. Once again, like our other cell types, our white blood cells and red blood cells, it all starts with the hemocytoblast again, this common stem cell. And then from there, the hemocytoblast will go through the developmental pathway, and eventually the stem cell will develop into what's known as a megakaryocyte. And this is the precursor to eventually platelets being formed. So the megakaryocyte that you can see here, right here, eventually this breaks down, and these small fragments, almost like shattered glass per se, become the platelets. This entire developmental process is regulated by a very important hormone known as thrombopoietin. Okay? Thrombopoietin. Sometimes when you get your blood, you're going ahead and you're getting your blood taken, blood chemistry analysis done, and you look at the various cells on there. You may not see the word platelets because sometimes platelets may also be indicated as thrombocytes. And thrombocytes is referring to platelets. The case of thrombopoietin, that hormone that you see here, the thrombo is referring to the platelets. Okay, so time for some more review here. So see if we're picking up on some of these concepts. So number 4, I should say. What is the function of platelets? What is the function of platelets? So what did I say here? We'll give you a little bit of time here to look over these choices. All right, and the correct answer here is letter D, blood clotting. So hopefully you put that as an answer, blood clotting. All right, our next review, number 5. Which of the following white blood cells are granulocytes? Which of the following white blood cells are granulocytes? So go ahead and take a look at your choices and go ahead and go ahead and select what answer you think is correct. All right, and the correct answer here is choice letter E. So I really hope you put that down, both B and D. Neutrophil is a granulocyte, likewise a basophil, and of course the one I don't have mentioned here is the eosinophil. And of course monocytes and lymphocytes are classified as the agranulocytes. Number 6 for review. This is the largest leukocyte and has a kidney-shaped nucleus. It also undergoes phagocytosis as its main function. So based off of these choices, what would be the correct answer that you would go with here? All right, and the correct answer, I would select letter C, the monocyte. So monocyte is the correct answer here. Number 7 for review. All blood cells are derived from a blank. What was that term I mentioned earlier in lecture? All right. And the correct answer here would be choice letter C. So letter C is the correct answer. All right, moving on. We're now going to take a look at the blood clotting process. And the term for that, you can see here, is hemostasis. So hemostasis is referring to that blood clotting process. Fast series of reactions for stoppage of bleeding. There are three major events related to this process of hemostasis. Stage 1, referring to vascular spasm or spasms. Stage 2, the platelet plug formation. And stage 3, coagulation. Really focusing on the blood clotting. So we've all encountered this before. Anytime you've had an accident, you know, are probably bruised or cut for sure. And this could be anything from a slight paper cut to maybe some cuts on your legs from a bike accident, where you hit some rocks, or maybe even more fun, maybe you get a fishing hook stuck in your thumb, which is always a blast, let me tell you. So now we're going to take a look at the events of hemostasis. So what is going to be involved with knowing these steps? So step number 1 of hemostasis is the vascular spasm. So you get the cut, you have the fishing hook stuck in your thumb, and there's blood everywhere, and it's really painful, and you got to go to the ER and get that pulled out, right? So this is happening definitely during the first 30 minutes of the injury. The cut in the smooth muscle lining of the capillaries, or whatever it may be, and this is causing vasoconstriction. This is important for narrowing of the blood loss, and eventually you will feel some pain. Step 2 to the process here is the platelet plug formation. So we do have the cut in the smooth muscle lining of the capillary, or maybe a tiny small vein perhaps. And this ultimately, there's exposure of collagen fibers, and ultimately then you're getting platelets to the vicinity. The platelets release chemicals, and these chemicals are platelet factors. So there's a variety of platelet factors that play a role here. And ultimately this leads to these platelets being sticky, and they ultimately plug the hole. So the platelet plug is what it's referring to, forms, plugging that hole in the lining of the smooth muscle. And then step 3 is the coagulation. This is where the actual clotting will occur then. You're going to have ultimately a fibrin, which forms a mesh, and that's going to trap red blood cells and platelets, forming the clot. So there's many steps involved in coagulation, many chemical reactions occurring. And ultimately, fibrin is the end product that's formed. Okay? So that's known as fibrin. This forms the basis of the clot, which is pretty important to know. Here we're taking a look at a very simple diagram looking at the 3 main phases of coagulation. It may look really crazy, but your textbook actually goes into a lot more in-depth look at it. But I've tried to make it as simple as possible. So during phase 1, there's many chemical reactions occurring, and you ultimately form what's known as a prothrombin activator. This activator, as the term is referring to, will activate this plasma protein, prothrombin, here in phase 2. And in turn, will go ahead and form thrombin. Thrombin is a very important enzyme, okay, which is key for blood clotting. And then phase 3, we start with what's known as the fibrogen. So this is a very soluble protein clotting factor. And this critical enzyme, thrombin, will help to go ahead and get this chemical reaction going to assist ultimately in forming our fibrin here. And this is our end product here, is the fibrin mesh that's cross-linked. Insoluble protein, fibrin strands. And again, thrombin, this critical enzyme here. And if you remember what an enzyme stands for, it's a biological catalyst that helps to speed up chemical reactions. This picture is pretty sweet. This is looking at red blood cells that are all kind of connected together because of the fibrin mesh. So all this brown material here is the fibrin. All through here, all through here. And it almost looks like a fishing net is one way to look at it, a spider web. So important to hold those red blood cells in place until the healing has occurred. But eventually, unneeded clots need to be removed. And that's the idea of the overall fibrolysis. The removal of unneeded blood clots when healing has taken place, which begins within the first couple days. But one type of disorder that's out there in the population, and maybe you've heard of it before, is hemophilia. So hemophilia is referring to a bleeding disorder. And unfortunately, it's genetically passed down from one generation to the next. And usually is found more likely than not in males versus females. And a lot of that, unfortunately, is because of the idea that males are XY, females are XX, and it's an X-linked recessive type of disease. Symptoms typically here are prolonged bleeding around joint cavities for sure. And this can start very early in life sometimes. So parents typically can pick up on this in their small children. The treatments that are out there, there are plasma transfusions that can be taken. And of course, injection of missing clotting factors. And sometimes that's the case. The child is born with missing factors, and so they're going to need regular injections of those blood clotting factors. We're now going to take a look at blood clots. There's certain different types of blood clots found around the body, and they can be very dangerous. They can lead to many cardiovascular problems. So we look at the terminology related to these blood clots. The first one we're going to take a look at in here today is known as a thrombus. So the thrombus is referring to a clot that develops and will persist in an unbroken blood vessel. And this blood clot can start anywhere, this thrombus. So it could be a blood clot in the leg. It could be a blood clot up in the brain. If it's in the heart, that's pretty severe because that can definitely lead to a heart attack. So definitely not a good situation to have the thrombus. Another type of blood clot is known as the embolus. So this could be extremely dangerous. So maybe you start with a blood clot in the leg, and the blood clot is mobile and it can move. And maybe the blood clot moves from the leg up to the brain, and then we're talking about a stroke. Or maybe it moves to the heart, and then we're talking about a heart attack. Or maybe it goes right to the lungs, and then we're talking about pulmonary embolism. So pretty dangerous for sure. And there's plenty of risk factors for developing these clots. A big risk factor, of course, is smoking. So someone who's smoking cigarettes, that's a big risk factor, is smoking. So that's a biggie there, smoking. Sometimes it could be related to someone's diet. If they have a poor diet, they're obese, Definitely a big risk factor for developing blood clots is the poor diet and lack of exercise. Smoking's a big one. And of course, blood clots can be formed because of significant stress in the body. And stress can do a lot of damage to the body. So those are just some risk factors there. Of course, high blood pressure definitely is a risk factor as well. So high blood pressure too. There are a variety of different drugs that are out there to try to go ahead and eliminate blood clots, definitely reducing the chance of that happening. A very common one that a lot of elderly take is aspirin. So you can easily go to Walgreens or Myers or wherever and buy your aspirin. So that's a very common medication that someone can take simply. More in a clinical setting is the blood clot medication heparin. So a lot of cardiac patients can receive this. Definitely for those receiving a blood transfusion during more of a severe surgery will receive this. So that's a very important one there. And then another one is known as warfarin. And this is another nice important blood thinner here to reduce the blood clot. So this is given to a lot of people that are prone to having a stroke, especially individuals that have AFib, atrial fibrillation, are typically given the medication warfarin. So those are all some examples out there. So definitely be aware of these blood thinners for sure. One of the last items that we're going to review in this chapter today is looking at blood types and blood typing. So, blood typing, ultimately we look at the ABO blood grouping here. And when we look at blood types, there's type A, type B, type AB, and type O. And then, of course, we have to recognize that there's also, of course, there's A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative. Someone classified as being the universal recipient is someone who's type AB. And this is an individual that is able to receive all the other blood types. So it's pretty beneficial to be type AB. And then someone who is type O is classified as the universal donor. So they're able to donate their blood to all the other groups. The Rh factor is another critical component when looking at blood typing. First identified in rhesus monkeys over in Africa. The Rh factor determines if the blood type is positive or negative. So I kind of showed that on the last screen. The majority of Americans have a positive blood type, 85%, while negative is 15% approximately. There definitely can be some incompatibilities with the Rh factor, especially during pregnancies with women or even blood transfusions, and we'll talk about that a little later. So this table here is looking at the overall percentage of the U.S. population that have a certain blood type. So type AB, we mentioned the universal recipient again. You can see it's pretty low percentage-wise. And here we're looking at an erythrocyte, and we're seeing here what are known as surface antigens. So when we look at an antigen, this is a new term here, the antigen is referring to anything that the body perceives as foreign, even a mismatch red blood cell type. So blood type. So here we can see surface antigens here, A and B. And then there's a term here known as plasma antibodies. And these antibodies act against red blood cells carrying the foreign ABO antigen. So surface antigens here, A and B, and no plasma antibodies here. So someone that's type AB can receive A, B, and O. And then we have type B. You can see a little higher in the population, and definitely with certain ethnic groups you can see. They have surface antigens for B, but not A. So there are plasma antibodies that can be built up for A. And of course, this blood type will attack the type A surface antigen. So someone who's type B can only receive type B and O blood. Someone who's type A, higher percentages definitely we find in the US population. So surface antigens here for A, but not B. Antibodies can be built up for B. So someone who's type A cannot receive type B blood. They can have A and O, however. And then someone that's type O, very high in the population here we can see with individuals. And no surface antigens here. Plasma antibodies built up for A and B. And someone that's type O can only have type O blood. Now another component that this chart doesn't show is positive and negative. So one thing to consider here, if someone has a positive blood type, so if someone has a positive blood type here, so positive blood type, they can of course receive the positive blood type, but also the negative blood type. However, if someone, and we'll erase this, if someone had a negative blood type, they can only receive negative blood. They can't receive positive blood. Okay? So that's something to definitely make sure to remember. So someone who's type AB and they're AB positive, they can receive all these positive types here, but also the negative. In contrast, if I just go down to type O for an example, if someone's the negative, they're O negative, they can only receive O negative blood. If they were O positive, they can receive O positive and O negative blood. So, important to remember. So again, someone has positive blood type, they can receive the positive blood type and the negative blood type. But if someone has a negative blood type, they can only receive negative. So, maybe you did a blood lab in 152, maybe not. But one type of lab that can be performed is using a blood kit that has depression wells in it. And the depression wells, we're referring to these circles here that are depressed down slightly. And maybe you're working with an unknown blood type. You have no idea what type it is. And so what can happen to identify the blood type is to go ahead and add these various serums. So here we have an anti-A serum. Here we have an anti-B serum. Here we have an anti-RH serum. And the anti-RH overall serum here ultimately will determine if the blood type is positive or negative. These antibodies, of course, referring to type A or type B blood. So we have an unknown here, and these serums are added: anti-A serum, anti-B serum, and anti-RH serum. And the one thing to make sure to keep a close eye on is looking at the clumping of blood, which is termed the agglutination. So agglutination is referring to the clumping of blood. The antibody will reveal the presence of the antigen. So these serums are referring to these two antibodies. So in this well, the blood will clump in this depression well. And when that blood clumps, that's indication of O positive. Blood did not clump with the anti-A or anti-B antibody serums, so we know it's definitely not A or B blood. But because it clumped here, we know it's O positive. In this example here, you notice we have clumping of blood with the anti-A serum, but not with the anti-B serum or the anti-RH serum. We know the blood type is A negative. And finally, down here, we can see that there's no clumping of blood here with the anti-A serum, but with anti-B and with the anti-RH serum, the blood type here is B positive. And finally, our example on the bottom, we can see with the anti-A serum, clumping of blood down here, clumping of blood with the anti-B serum, no clumping of blood down here with the anti-RH, so we know this blood type is AB negative. Here's just a picture visually seeing the serums anti-A and anti-B. And when it comes to the clumping of the blood here, we can see that here, the agglutination. So clumping of blood here, clumping of the blood here with the anti-B serum. So obviously type AB. Here we have clumping of the blood with the anti-A serum. So type A blood. Clumping of blood here, so indication of the type B blood, and no clumping of blood here, so we know that's type O. All right, so here are some more review questions related to identifying an unknown blood type. So, kind of using this picture as a little bit of a guide, imagine that no agglutination occurred in all three wells. Again, these are considered wells. Imagine there was no agglutination in all three of the depression wells. What is the blood type? So no agglutination in all three wells. What would be the blood type? All right, and the correct answer, I would go with type O negative. So we have no agglutination in all three wells. You can see that one was O positive. Imagine there was no agglutination here. It's just, spots like this. Easily, that can be changed to O negative. Okay? How about another example? So number 9. Here we have agglutination in all three of the depression wells. What is the blood type? All right, and the correct answer here, I would probably go ahead and select letter B, type AB positive. So type AB positive would be the correct answer. So if you look here, this is AB negative. We could easily start clumping the blood here, just like this. Da da da, la 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 la. Yep. And fix that, and bam, now we have AB positive. So, very important concept to make sure to know. Definitely review this. We're now going to take a look at some examples of where we have an incompatibility of the blood type. Definitely here referring to an indifference with the Rh factor. So the first example we have here is an incompatibility with the blood type between mother and baby, or per se fetus. And this is known as the hemolytic disease of the newborn, also known as erythroblastosis fidalis, referring to the red blood cell here. Erythroblastosis referring to, unfortunately, destruction of the actual cell, and this is referring to the fetus here. So again, this will occur with an indifference of the Rh factor between mother and the fetus. And so as a preventative measure, there's testing, of course, of blood that can occur within the the placenta to determine that blood type of that fetus growing. And so one medication that the mother can take, ultimately that will play a role with the fetus, is a medication known as Rogam. Maybe you've heard of this med before, Rogam. And this is important to prevent the antibodies built up in mother to go after these cells of the growing fetus. And that's a great way to prevent destruction, a miscarriage of the child. Hopefully the child's not born dead or very anemic or jaundice or whatever it might be. So this medication is critical for a mother that has this indifferent blood type, Rh factor. So let's go through a story analogy here to help us understand this situation. So here we have new parents here. So here we have Adam and here we have Sally. We note the blood type here for dad is Rh positive and for mom Rh negative. So there is that possibility that the baby could end up having Rh negative. Or the baby, will be Rh positive. So that would be an imbalance between mom and baby. Rh negative for mom, Rh positive for baby. And this condition, for the most part, only occurs when the mother is Rh negative. And so here we can see the first pregnancy here. And mother is pregnant with the firstborn. Remember, mother Rh negative, fetus Rh positive. And it is possible, giving birth, there could be some blood that could ultimately mix. The pregnancy was successful after delivery. So the first child is born. And remember, there's no memory cells built up. So for the most part, typically that firstborn is healthy. Mother, Rh negative, that firstborn Rh positive. But because some blood did mix between mom and baby, it's possible that antibodies can build up and there could be complications in the second pregnancy. So mom and dad decide to have another baby, and here's the second. If mother does not take the medication, the Rogam we mentioned before, right? If she doesn't take it, then these antibodies built up from the foreign blood type in mother can directly go into the fetus and attack the red blood cells of the fetus and could lead to a miscarriage occurring or significant complications in the child if born. But if mom does take the medication Rogam, then that eliminates that possibility and that second child can be born and everything's fine. Again, a difference with the Rh factor, mom Rh negative, fetus, per se baby, Rh positive. And the last incompatibility we look at with the Rh factor, or simply someone's given the wrong blood type, they're supposed to have A, they got B, whatever it might be, is the blood transfusion that went wrong. So very important to obviously make sure your patient receives the proper blood type that they may need during surgery, etc. You would think these mistakes don't occur with everything being typed up with Epic's the typical program that's utilized now. But mistakes can still occur if someone's in a rush. Always double check. So what happens here is the individual is given the wrong blood type, and agglutination may take place, clogging vessels, perhaps in major organs like the kidney, lungs, heart, and this ultimately can lead to the rupturing and release of free hemoglobin in the bloodstream. And that can be dangerous because this could lead to renal failure. So we can see that here. Surface antigens on these red blood cells, opposing antibodies go after. You have significant agglutination taking place, and then hemolysis. Lysis refers to cell splitting. And ultimately, in time, all this hemoglobin going throughout the blood can lead to renal failure and death. So very important that the patient receives the proper blood type. And I know you guys will do an excellent job making sure your patients receive that blood type. And one final review question for you here today, number 10, referring to blood. So let's say you have a patient, his name is Johnny, and he has a gunshot wound to his left leg. It was pretty severe, maybe it severed several arteries and needs a blood transfusion. His blood type is A positive. What type of blood can he receive? So what would you recommend for Johnny? So go ahead and take a couple seconds here, minute or so, and look over this and see what would be the correct answer here. Okay, and the correct answer I would go with, if Johnny's A positive, I would definitely select E, all the above. Johnny can have all of them. Obviously type A, but he can have type O too, the universal donor. And if you have a positive blood type, you can also have a negative blood type. So all of these would apply to Johnny. All right, so that's the first lecture down, and I hope you enjoyed today. And until next time, we leave you with a great song that was played, of course, when Michael Jordan came out on the court to play for the Chicago Bulls.